So, months into the CV lockdown, millions of people lost their jobs, all kinds of insanity going on. People worried about, are we going to have a W-shaped recovery instead of a V-shaped recovery? And in the midst of all this, Fauci comes out and tells everybody, if you've had the common cold in the past 5 years, your T cells will most likely protect you from CV. Let's get into it. Dr. Fauci says this may already be keeping you safe from CV. A common cold from 5 years ago could be helping you fight the CV, Fauci says. One of the biggest questions amid the CV-19 pandemic has been, why does the virus kill some people and leave others without any detectable symptoms? Now, 6 months into the pandemic, we are finally getting some answers. According to Anthony Fauci, MD, director of National Institute of Allergy and Infectious Diseases, if you are someone who's known to get the common cold year after year, you could have some protection against the novel CV, thanks to the T cells in your immune system. If you look at your immune system metaphorically as an army with different levels of defense, the antibodies prevent the virus from getting in. So that's kind of like the first line of defense, Fauci told McClatchy in a recent interview. For those viruses that do escape and infect some cells, the T cells come in and kill the cells that are infected or block them. Fauci said much of the research on CV has been focusing very exclusively on the antibody test. But, he said, T cells are an equally important component of the immune system. Well, no shit. You don't say. Because CV-19 is a new CV, it was initially believed that your T cells would not be able to detect it. It was assumed that T cells would only be found in people who have already had CV-19. But a new National Institute for Allergy and Infectious Diseases study published in the journal Science on August 4 suggests that up to 50% of people who have not been exposed to the CV have T cells needed to fight the virus. Similarly, a German study published in the journal Nature at the end of July looked at 68 healthy people who had not yet been exposed to the CV. Among them, 35% had T cells in their blood needed to attack the novel CV. As a result of this new research, experts believe that healthy individuals may have generated these T cells when fighting similar infections from related CVs in the past, like the common cold. And the more recently an individual was infected with another type of CV, the greater the chances are that they have some protection from CV, Fauci told McClatchy. It's sort of like a 1-2 punch, he said. It's conceivable that the T cells that you've made in response a couple of years ago, 3, 4, 5 years ago, when you were exposed to a relatively benign CV that causes the common cold, could actually hang around, and when you are exposed to SARS-CoV-2, could have some degree of protection. Serving as a secondary line of defense in the immune system, once antibodies have failed or faded away, T cells also last much longer than antibodies. So if you've read those startling reports that antibodies don't last, like a July study out of the United Kingdom that found CV antibodies declined after just 3 weeks from the start of the infection, worry not. These reports ignore the role of T cells, according to Fauci and other experts, which is just as pivotal. Mesha Dolja, MD, senior scholar at Johns Hopkins University Center for Health and Security, told CNN that pre-existing T cells may also help us understand why CV affects people so differently. If you could compare people maybe with severe and mild illness and try and look at the T cells in those individuals and say, are people who have severe disease less likely to have cross-reactive T cells versus people who have mild disease maybe having more cross-reactive T cells? I think that there's biological plausibility to that hypothesis, he said. It's clear, though, that the T cell presence doesn't prevent people from getting infected, but does it modulate the severity of infection? That's what it appears could be the case. So let's recap. They push the numbers up, doing everything that we've covered. They don't even hide the average age of death. An average number of comorbidities. They're not hiding any of this. And now they're telling us if you've had the common cold in the past 5 years, there's a possibility, a large possibility, that's why there are so many asymptomatic carriers. Because we already have the T-cells program to fight the cold, because it is a cold, and it's always been a cold. And there's links to the scientific studies that are cited in this article. I don't feel like going into them anymore. Honestly, I don't even feel like covering this subject anymore. But it is what it is. At risk of getting off topic here, but did you guys know that Kamala in Finnish means horrible? And so you add this study to that study, and because that study says if you've ever had a cold in the past 5 years, you most likely have the T cells to fight off the CV. You don't have the antibodies to prevent it from entering your body, but once it gets there, you have the T cells to kill it. This is why we have so many asymptomatic carriers, as they're called. But if you take into account what they're saying in that article, and then you come over here to read this study, this was a study on 455 contacts, which aimed to study the infectivity of asymptomatic carriers. The groups were divided into 35 patients, 196 family members, and 224 hospital staffs. We extracted their epidemiological information, clinical records, auxiliary examination results, and therapeutic schedules. The median contact time for patients was 4 days, and that for family members was 5 days. So these people were in contact with asymptomatic carriers for 4 and 5 days. So how many got the virus? In summary, all the 455 contacts were excluded from having SARS-CoV-2. None of them contracted it. 455 people studied with an average time of 5 days in a house with an asymptomatic carrier, and none of them got it. So we know the average age of death. We know the number of comorbidities. We know they moved people into nursing homes. We know that most people are, quote, asymptomatic carriers because they have T cells that protect them from this virus that they developed from the cold. We know that asymptomatic carriers are not spreaders of the virus. We know that it doesn't infect children at any kind of statistical significant rate. We know all of this. On top of that, we know that they've fudged the numbers. We know that they've hidden treatments. We know all of this. And why have they done all of it? They've destroyed the economy. They've forced us to lock down. They make us walk around with pieces of paper on our face as if that is some kind of protection. Why? Did you know that there's a difference between the vaccine that Moderna is coming out with and the typical vaccines that people get? I don't want to get lost in the weeds here on vaccines, but let's just do a generality, right? So in the past, the objective is to inject you with either a non-live culture of the virus you're trying to vaccine for, with something that will cause an immune reaction like thimerosal, or to inject you with a small amount of the actual live virus, and that would cause you to develop the antibodies against said virus. This is the methodology behind your typical vaccines, and there's been all kinds of arguments about which component we should use to cause their immune response and which cell lines we should grow the cultures on, and there's been all kinds of different things going into that I've covered on the channel. But Moderna's never made anything for human consumption, and they're making a vaccine that's never been given to humanity. Ever. So what's the difference? Well, let's read an article here. As Moderna's CV vaccine takes the lead, its chief medical officer's recent promotion of, quote, gene editing vaccines comes to light. Moderna's chief medical officer has described the company's products as, quote, hacking the software of life and permanently altering a person's genetic code. If Moderna is poised to bring the first CV vaccine to the market, a deeper look at his comments and his employer are warranted. More and more frequently, government officials, political pundits, and self-appointed global health experts like billionaire Bill Gates have been instructing the public that mass gatherings and any semblance of normalcy will not return until a vaccine for the NCV is created and subsequently distributed to the masses. In recent weeks, it has quickly become apparent that the leading CV-19 vaccine candidate is the messenger RNA vaccine being developed by Boston-based Moderna Incorporated. Today, Moderna announced that its vaccine candidate, named mRNA-1273, appeared to produce an immune response in 8 people who received it. Whoa! Moderna's response is odd given that the study in question is focused on safety and is actually not designed to measure effectiveness of the vaccine, according to a report in Time. Notably, none of the study's findings on vaccine safety were reported aside from claims it was generally safe. It is also worth noting that this safety-focused study only began in March and thus, to date, represents only an examination of the vaccine's efficacy in the very short term. Major media outlets in multiple countries ran with the headlines trumpeting that Moderna's CV-19 vaccine shows promising early results and has presented encouraging early signs because of its purported ability to produce CV-19 antibodies in humans. In addition, these media reports failed to raise other simple yet necessary questions, such as how a sample size of only 8 people can translate into scientific findings of any real significance without further testing involving larger sample sizes. They also failed to note that the study in question is not even finished, as a U.S. government press release noted that the findings in question are merely interim results. In addition, the study is being led by the U.S. National Institute of Allergy and Infectious Diseases itself, headed by Dr. Fauci, who is a key figure in the U.S. government's coronavirus response. Fauci's up to his neck in it with Moderna and Remdesivir, the company that makes Remdesivir, so, which is not that deep in with them because it's just up to his neck and he's a little guy, but he's up to his neck in it. Though it is unclear if these encouraging early signs will be replicated in future tests of larger samples that are actually designed to test the vaccine's effectiveness, the news is surely welcomed to Moderna, given that their past mRNA vaccines failed to produce hardly any immune response at all, explaining why the company has never brought an mRNA vaccine to the market in its entire history as a company. However, since at least last fall, Moderna has sought to resolve this issue by adding nanoparticles to its mRNA vaccine, a modification financed by the Pentagon's Defense Advanced Research Projects Agency, otherwise known as DARPA. So get this, last fall, before CV had ever been spoken of, they were working with DARPA to create nanoparticles to go inside of our CV vaccine. Isn't that awesome? These guys, they think ahead. They love us so much. Moderna is a strategic ally of DARPA and has received millions from DARPA and the Bill and Melinda Gates Foundation several years prior to the current coronavirus crisis. DARPA's plans for nanoparticles and nanotechnology and their potentially Orwellian applications were the subject of a recent The Last American Vagabond report, and I'll put all the links in the description as usual. Thanks to the interim results of this new study, Moderna is set to take the lead in the race to gain government approval for a CV-19 vaccine. Moderna had already pulled ahead of other CV-19 vaccine candidates in recent weeks, being the first vaccine in the U.S. to go to human trials after it was allowed to skip animal trials, and also enjoying strong support from the U.S. government. For instance, Moderna's CV-19 vaccine recently received fast-track approval from the FDA after receiving the green light to proceed to Phase 2 testing prior to the results of Phase 1 being published. Isn't that nice? Dr. Stephen Hoge recently said that the company now expects to begin the final third phase of testing sometime this summer. Who are these people that are going to let them inject them with this substance? In addition to support from the FDA, Moderna has also received considerable U.S. government funding, $438 million from the Biomedical Advanced Research and Development Authority, BARDA. So we got DARPA and BARDA, a division of HHS overseen by HHS's Assistant Secretary for Preparedness and Response, ASPR, Robert Kadlec. So we have DARPA, BARDA, and ASPR. Moderna has also stated that it is directly collaborating with the U.S. government to bring its vaccine candidate to market. Moderna's considerable lead has been the result of backing that it received in January from the Coalition for Epidemic Preparedness Innovations, CEPI, which was founded in 2017 by the governments of Norway and India, along with the World Economic Forum and the Bill and Melinda Gates Foundation. Moderna's CV-19 vaccine has also received additional millions from the longtime Moderna backer Bill Gates. Gates recently authored an article where he described Moderna's mRNA vaccine for CV-19 as the most exciting and discussed it at length. Yeah, he also put out an op-ed today saying that he was calling for an end-to-end encryption. So he wants to get rid of end-to-end encryption. No more VPNs for people. No more WhatsApp. No more Signal. He doesn't like that. He doesn't like people being able to talk without him getting his grimy hands on it and his long lizard tongue. I guarantee you that man has a long tongue. Gates' affinity for Moderna may owe to the fact that Moderna's co-founder, MIT's Robert Langer, is a Gates associate whose lab developed the Gates-funded quantum dot tattoo vaccine identification marker that is, quote, visible using a special smartphone camera app and filter, and was described by Science Alert as a low-risk tracking system. Another Langer-Gates partnership is a birth control microchip inserted to the body that releases contraceptives and can be turned on and off wirelessly. Gee, I wonder if it has Plan B in there with it. Meet Dr. Zax. With Moderna taking a firm lead relative to the other CV-19 vaccine hopefuls, it is worth taking a closer look at the man who has overseen its development. Moderna's current chief medical officer, Dr. Tal Zaks. Zaks, an Israeli citizen who began his career at GlaxoSmithKline, oversees preclinical development, clinical development, and regulatory affairs for Moderna and all its subsidiaries. In a 2017 TED Talk, 2 years after joining Moderna, Zaks spoke at length about how he views mRNA vaccines and their modality, including those he produces at Moderna, in a speech entitled The Disease Eradicating Potential of Gene Editing. Zaks' description of Moderna's mRNA products as making permanent edits to human genes clashes with often touted claims that the genetic material in mRNA vaccines degrade over time and do not permanently alter human genetics like DNA vaccines. Beginning his talk, we're just going to watch the talk. The summary of Zach's talk encapsulates his view as the following simple question. If our cells are the hardware and our genetic material the operating system, what if we could change a few lines of code? Seemingly suggesting that the permanent introduction of changes into the human genome is as simple as troubleshooting or programming a computer or phone application. It also says that Zaks considers the future of personalized medicine to be gene editing vaccines tailored to each patient's immune system. The TED Talk recommended after viewing Zach's speech on the TED Talk website notably broaches a key point that Zach's overlooks, namely that gene editing can change an entire species forever. Zach's statements are noteworthy and concerning for several reasons, including the fact that DARPA, Moderna's strategic ally, is also openly funding research aimed at reprogramming genes and manipulating genes or controlling gene expression to combat viruses and help human bodies withstand infection caused by CV-19. The DARPA-backed project would use a method that is known to cause severe genetic damage that has actually been shown to aggravate the conditions it was meant to cure. With such permanent gene-altering technology on the fast track to become the first CV-19 vaccine widely available for use, it is deeply concerning that this experimental vaccine with potentially far-reaching consequences is being rammed through thanks to fervent support from both the U.S. government and controversial philanthropists that apparently have little interest in studies examining mRNA vaccines' long-term effects. On July 16, 2018, Scientific America released an article entitled Potential DNA Damage from CRISPR Seriously Underestimated Study Finds. I'm going to link all this in the description. I suggest that you guys go down here and read these articles. The way that they're thinking that they're going to design these vaccines is essentially like thinking about jamming a bunch of ones and zeros into an envelope and stuffing it into our genetic code and expecting that to create perfect code moving forward. It's absolutely heinous. And then this is childrenshealthdefense.com. It's RFK Jr.'s webpage. Despite Moderna's cheery press release this week, the clinical trial result for its groundbreaking CV vaccine could not be much worse. The vaccine, developed and championed by Anthony Fauci and financed by Bill Gates, used an experimental mRNA technology that the two men hoped would allow rapid deployment to meet President Trump's ambitious warp speed timeline. Dr. Fauci was so confident of his shot's safety that he waived ferret and primate studies. Moderna suspiciously reported no specific health data from its mouse studies. That appears to have been a mistake. 3 of the 15 human guinea pigs in the high-dose cohort suffered a serious adverse event within 43 days of receiving Moderna's jab. Moderna did not release its clinical trial study or raw data, but its press release, which was freighted with inconsistencies, acknowledged that 3 volunteers developed grade 3 systemic events defined by the FDA as preventing daily activity and requiring medical intervention. Moderna allowed only exceptionally healthy volunteers to participate in the study. A vaccine with those reaction rates could cause grave injuries in 1.5 billion humans if administered to every person on Earth. That is the threshold that Bill Gates has established for ending the global lockdown. Moderna did not explain why it reported positive antibody tests for only 8 participants. These outcomes are particularly disappointing because the most hazardous hurdle for the inoculation is still ahead, challenging participants with wild CV infection. Past attempts at developing CV vaccines have always faltered at this stage, as both humans and animals achieved robust antibody response, then sickened and died when exposed to the wild virus. Moderna's press announcement heralded positive interim phase 1 findings. I have forwarded that claim to my colleagues in securities law. FTC rules restrict the amount of lipstick public companies may slather on bad donkeys. And this is a link to a Cambridge University website that gives a good introduction to what an RNA vaccine is, or what it will be, because it's not something yet. So what it would be. Like I said, guys, I'll have links in the description or down in the comment section for all this information, and I'm going to leave you with this guy, the head of Moderna's TED Talk. I suggest you watch it. It's highly enlightening. But anyways, guys, I'll catch you next time. Like, share, sub. I'm out of here.
Since, as I've trained as a physician scientist, we've been living this phenomenal digital and scientific revolution. And I'm here today to tell you that we're actually hacking the software of life, and that it's changing the way we think about prevention and treatment of disease. So here's all the biology you need to know in 30 seconds. Our body is made out of organs. Our organs are made out of cells, and in every cell, there's this thing called messenger RNA, or mRNA for short, that transmits the critical information from the DNA, our genes, to the protein, which is really the stuff we're all made out of. This is the critical information that determines what a cell will actually do. And so we think of it like an operating system. And it's not just in every cell of our body, it's actually in every cell of every organism alive. It's the same thing. And so if you could actually change that, which we call the software of life, if you could introduce a line of code or change a line of code, it turns out that has profound implications for everything from the flu to cancer. And I'm going to demonstrate that with 3 short examples. Let's start with the flu. So many of us get a vaccine. What is a vaccine? It is an injection in our arm where we get bits and pieces of the virus, the proteins, and that teaches our immune system to recognize the virus, and so when we get infected, we're not sick. Now imagine if instead of giving the protein, we would give the instructions on how to make the protein, how the body can make its own vaccine. That's an mRNA vaccine, and here's what it looks like from the cell. So the traditional approach has protein floating around your cells. An mRNA vaccine approach has the cells themselves in your own body making the vaccine. What's more alarming, a stranger prowling the neighborhood or somebody who just broke into your ground floor and tripped the alarm? That's what happens with an mRNA vaccine. You've tripped the alarm wire, and now the cell is dialing 911. It's calling the police at the same time as it's making the protein and saying, that's the bad guy. That's how an mRNA vaccine works. And for the last several years, we've shown this actually works in a whole multitude of animal models. Earlier this year, we published the first actual study in people. And it actually works in people. We took a group of volunteers and injected them with a messenger RNA vaccine against a variant of influenza. And all of these volunteers got the immune response we were hoping to see. The side effect profile was pretty benign, what you would see with any normal type vaccine. So we've proven the principle. This actually can work. It works in people. And now we're going to be developing a whole slew of vaccines against diseases for which we don't have one. So that's infectious disease. Now, for the second example, let's talk for a minute about cancer. Horrible disease. Cancer has affected the lives of many of us and will affect the lives of many more of us as we age. The problem with cancer at the cellular level is that the DNA is screwed up. You've got these mutations, and this screwed up DNA leads up to screwed up information that makes screwed up protein. And so the cell loses control. Now, how do you figure out what is actually screwed up? Well, you got to figure out the whole sequence, right? It took us decades and billions of dollars to sequence the human genome. And we've done that. We achieved that in 2003. And now we're less than 15 years later, and it takes us a week. And we can do it for every patient. So now we can go and figure out what exactly is screwed up in a patient, and we can use that information to make a vaccine. We take that information, say a patient with lung cancer, and we take the biopsy, we figure out the sequence, we figure out their immune system, and that all becomes information. It goes up in the cloud into a bioinformatic algorithm, and then automatically makes a vaccine that we administer into their normal tissue, into the muscle, to try and wake up their immune system. Now, the challenge, of course, is that every person's cancer is different. Mutations happen by random chance. And so to do this, you have to make it personalized. So this is me, but if every patient is different, what we're going to have to do is make a personalized cancer vaccine for every patient. And that's exactly what we've started to do. Every patient gets a vaccine that's based on the sequence in their own tumor. So when we started to do this a couple years ago, my CEO stopped by one evening and said, "Tal, I get the idea, but is this going to work?" And I said, "Look, Stefan, I don't know, but we've got all the pieces to try and answer the question, so we should try." And today, I can tell you that I still don't know if it's going to work, but I know we're able to actually run the experiment. Earlier this week, the first patient was treated with a personalized cancer vaccine we made just for her. Thank you. So in the months and years to come, we will know the answer of whether we can actually wake the immune system against somebody's cancer with a personalized cancer vaccine. So stay tuned. I'm going to finish with a third example of something called methylmalonic acidemia, or MMA for short. Now, the name doesn't matter. Okay, this is just a disease that is caused by an enzyme that's critical for metabolism. And children are born, and they lack this one crucial gene. And so their body is not able really to fight infection properly, or anytime they have any sort of stress, their body goes into crisis. They have one gene that's gone awry, and it causes a really significant disease. If you look at what happens over time for these children, about 1/3 of them don't make it to the age of 10. You see here the survival curve, whether the gene is completely lost or whether there's just an aberration in it, the survival is impaired. And what do? Well, there's not much you can do because the missing protein is actually missing inside their cells. So what do? Well, here's what we do. We take out their liver, and we transplant the liver from a donor that is healthy and normal into these kids. Think about it. They're missing one critical piece of information, and what we do is transplant an entire organ. Well, it fixes the problem, but what if there's a better way? What if we could fix the missing information? So based on innovations, nanomedicine, a new class of invention that Bob Langer across the river at MIT and Cambridge has been inventing, we're now able to package this information in messenger RNA with the goal of giving it as an infusion and then having it go to the liver to replace that missing information. Is this going to work? Well, we know the biology works. So together with the National Institutes of Health, we've studied this in a mouse model. And this mouse has been engineered to have the exact same problem that the kids have. They're lacking the same gene. And you can see in the red line what happens to these mice when they're born. Pretty much immediately, they die. They cannot cope with stress. But if you inject messenger RNA that codes for the one missing protein that replaces that information, these mice, all of them survive, as you can see in the green line. And if you look at them, they not only survive, they're actually growing, they're gaining weight, they look like they're healthy littermates. We're hoping to start the clinical trials in the near future, and the idea is the same thing here. If you think about what it is we're trying to do, we've taken information and our understanding of that information and how that information is transmitted in a cell, and we've taken our understanding of medicine and how to make drugs, and we're fusing the two. We think of it as information therapy. I started by telling you about Jonathan, and 30 years ago, when I was a nurse in the intensive care unit, I worked 2 night shifts. And Jonathan came in when he was about 12 months old and very quickly became dependent on a ventilator. And for the next 15 months or so, every time I came into the unit, he was my patient to care for. Bathe, feed, treat, play with. He couldn't talk. He was on a ventilator, but he was very much alive. And you could tell, you could play with him. His eyes would follow me after a while. He would recognize me. Until one day I came into the unit for my shift and he was no longer there. He had died because of an infection in between shifts. Imagine a world where we can not just diagnose, but we can actually use the information to create vaccines, to wake up the immune system to something like cancer, and to fix the missing information for children with diseases like Jonathan so that they can live the ICU and live a healthy life. Thank you.